Everybody. Welcome to our Translational Neuroimaging Education Program. I'm the moderator for this event today. My name is Riyanka, and I work at the Department of Psychiatry at the University of British Columbia. Before introducing our guest speaker today, please allow me 2 minutes to have a quick introduction of our program. So, this educational program is nested within the lifespan working group of the Enigma Consortium, which spans more than 45 countries and involve over 4,000 researchers worldwide. Our program is an inclusive program that welcomes researchers at all levels of neuroimaging skills and from diverse training backgrounds. The directors of the program are Dr. Shalala Haas from Mount Sinai, New York City, and myself, and we do have an international advisory committee. Please consider to join our mailing list if you wish to receive notification of updates about our program. We have a YouTube channel where you can find the recordings from previous events. I'm also recording today's event, so if you are interested, please keep an eye on our YouTube channel. We will be uploading it very soon. Just a heads up, we will only be sharing the presentation by our guest speaker, not the Q&A part. So please feel free to raise any questions for our guest speaker after this presentation. We're both on Twitter and Blue Sky, and if you are on either platform, we would love for you to follow us there. If you wish to share your work through this platform, please get in touch with us. We would welcome any type of collaborations. Today, we are very excited to welcome Yixue Feng, Wendy. Wendy is a PhD candidate in neuroscience at the University of Southern California, where she is advised by Dr. Paul Thompson at the Imaging Genetics Center. Her research focuses on diffusion MRI, using it to map both micro and macrostructural changes in white matter tract involved neurodegenerative diseases. Wendy is also passionate about developing and applying machine learning and AI methods for large-scale multi-site neuroimaging data to make analysis more reproducible and reliable. So in today's talk, Wendy will walk us through 3 key points. First, the streamline density normalization method, and she will show us a demonstration. Second, how SDNorm can enhance the reproducibility of bundle reconstructions and profiles. And lastly, the overall bundle segmentation pipeline, including quality control and visualization. We're really looking forward to learning more about this impressive new work. So Wendy, the floor is yours now.
All right, thanks so much. I'm going to share my screen. Oh. All right, can everyone see the screen okay?
Yes.
All right, perfect. Thank you, Dr. Ke, for that wonderful introduction. It is my pleasure today to be at the Translational Neuroscience program webinar today. Kind of going over some of the outline. I know you mentioned that earlier, but we actually will be starting with some background on diffusion MRI, tractography, and tractometry. So if you're not familiar with those, don't worry, we'll go over them in detail. And then we'll jump into streamline density normalization. And then finally, the bundle segmentation and quality control pipeline. So diffusion MRI is a specialized type of MRI that is sensitive to water diffusion in the brain. So in the brain's white matter, where you have axons that are these long tubular-like structure, water will typically flow along it. So using diffusion MRI, we can probe the white matter structure underneath. And tractography is the method that we can use to integrate these local diffusion orientation information to combine them into these long-range projections or pathways. And each individual, each line is called a streamline. And if we do this, all over the brain, we can get these whole brain tractograms. And from these whole brain tractograms, we can also segment into these different bundles or different collections of streamline that typically reflects different underlying anatomical structures, like the archaeofasciculus or the corticospinal tract. So one quick note on the terminology is that we typically use streamlines or bundles or tractogram to refer to the computational reconstruction of these different structures, but the underlying structure are typically referred to as fibers or tract or fasciculi. And a different kind of information we can get from diffusion MRI is these microstructure, and where we can model the fusion signal at each voxel level using these different models. One such model is a diffusion tensor model, or DTI, where if we use a tensor to describe the diffusion information at each voxel, it can describe from isotropic diffusion to anisotropic diffusion. And from these models, we can compute quantitative measures such as fractional anisotropy, which describes how anisotropic the diffusion is, or mean axial and radial diffusivity, which describes the magnitude of these different eigenvectors of the tensors. And these microstructural measures have been found to be sensitive to different diseases such as Alzheimer's and Parkinson's disease. So this study on the left here looks at a cohort of dementia subjects compared to controls, and they find there were widespread alterations in mean and radial diffusivity in particular in dementia subjects compared to controls. And this figure is from a paper that was published last year by the Enigma Parkinson's group, where they found this very interesting pattern of increase in FA in early stages of Parkinson's, followed by decrease in FA in the later stages. And this early increase in FA is sort of the opposite direction of what is typically understood of FA, where we expect FA to decrease in aging and neurodegeneration. But this kind of stage-specific analysis was able to highlight this pattern. And these studies typically use a voxel-based white matter analysis method called tract-based spatial statistics, or TBSS, which focus the analysis on these white matter skeletons that you can see marked in blue in this figure here. And this method came out in 2006, but has since been used in many of the Enigma studies. And these white matter skeletons can be further divided into different regions of interest, such as the Enigma DTI template shown here. And the Enigma TBSS protocol was the one that was used in some of these Enigma studies. And if you pay attention to some of these regions of interest that's shown here, these ROIs actually span quite a large areas of the brain. So in recent years, there have been some more interest in getting better specificity and localizing, where these white matter microstructure alterations are taking place. And one such method to do so is tractometry, or a long-tract mapping of white matter microstructure, which combines these different microstructural measures with tractography. So the idea of tractometry has existed for a long time, I think with some of the earlier studies going back to 2005. But it wasn't, excuse me, until recent years with better tracking methods as well as open source software being available, that tractometry has really gained more traction. And so one such method is automated fiber quantification, or AFQ, where this method provided a generalized framework for mapping these along-tract, for instance, FA differences along like 100 different locations along the tract. And they focus on more of these, the core central part of the tract where the streamlines are more uniform and coherent. And later on, 2020, bundle analytics, or BOAN, came out, and this was developed by Dr. Bramsh Chandio, who is a longtime collaborator and mentor of mine. And she developed this with the DIPI team, where they not only improved upon how these along-tract profiling worked by creating these, it's a little bit small here, but you can see this along-tract segments, but also provided a whole framework for doing group statistics using linear mix model, And more recently, she has also added combat harmonization that we'll talk about in a bit. And you'll see Buon being kind of mentioned later on throughout this talk as well, as it is a method that I use a lot in my PhD studies. So this is one study that we did using Buon to map along-tract white matter abnormalities and DTI metrics in dementia. And this was published in Alzheimer's and Dementia this year, where we looked at a cohort of more than 1,000 subjects from the ANI phase 3 data set. And this is also in collaboration with the National Institute of Mental Health and Neurosciences from Bangalore, India, as part of the Enigma India efforts. So we used Buon to look at, again, the DTI alterations in dementia subjects compared to controls. And we found, consistent with the prior studies, that there's a very widespread alterations in mean, radial, and axial diffusivity. But if you pay attention to some of these tracts, the patterns illustrated on the tracts, you'll see that the effects are not uniform along the length of the tracts. And this pattern is very prominent in for FA, where if you look at the projection fibers here, we saw this near the more distal part of the projection fibers near the cortical surface. There is a consistent decrease in FA, but in the more proximal areas of the projection fibers, we see this consistent increase in FA. So in a typical regions of interest analysis, this will not be possible because these signals will very likely cancel each other out. But tractometry is able to provide us with the specificity to be able to map out these patterns. Now that we have this wonderful method, the challenge that remains with scaling up, not just tractometry, but really diffusion MRI analysis in general, is this challenge of dealing with side effects and doing harmonization. So one of the most widely used method to do harmonization is combat, whose first introduction to neuroimaging was actually applied on DTI, where the idea is to have certain variables reflecting biological variability, if we want to preserve things like age or sex effects, but try to remove these batch effects that's due to scanners, site protocol, or processing strategies. And over the years, combat has really evolved into a whole family of methods, into different variants like combat GAM or even deep learning versions. And more recently, combat was integrated into the Buon tractometry pipeline. And this paper was showing that adding combat harmonization really was able to alter the statistics, of group statistics. And in this case, it actually boosted the sensitivity to detect dementia effects on the arcuate fasciculus, and which you can see from this p-value plot here. So even with these harmonization methods, the main goal is really to address the site difference in terms of how it affects microstructural measures. But there's another very important piece of the puzzle here, is that the same side effects and the same scanner effect that is affecting these DTI measures is also going to affect tractography. So this figure is from the Handbook of Diffusion MR Tractography that came out this year, which is a very wonderful resource. And they have a whole chapter dedicated to talking about the different sources of these, variability. And this is a figure that was showing the different variability in processing strategies. These very seemingly minor parameters like the max angle or the FA threshold can drastically alter what the tracts, the reconstruction look like. And this figure on the right here is a paper that summarizes how the choice of even the bundle segmentation pipeline, the scanner, the B-value, and the acquisition protocol can also affect the bundle reconstruction a lot. So this really highlights a need to kind of, in terms of how can we mitigate this variability in tractography and to improve the reproducibility of our tractography studies. So when it comes to tractography post-processing and evaluation in reproducing and in improving the reproducibility of tractography, a family of methods is committed to dealing, to kind of frame this as a tractogram filtering problem. And this type of method is applied on whole brain tractograms, where you sort of have a ground truth data to compare these tractograms with. So for instance, the method SIFT developed by the MRTrix team try to match the streamline density based on how well they match the underlying FOD signal. And this is the commit model, where they do a more complex model where they try to use microstructure to inform how we can filter a tractogram by modeling these different tissue compartments like intracellular, extracellular, and isotropic. While these methods are very nice, one challenge with that is these cannot be applied on the bundle segmentations. And the main reason for that is because of the bottleneck problem that's well known in tractography. This is a figure from a paper that summarizes phenomenon where they looked at not only at the voxel level, but even finer at the pixel level, which is a unit smaller than the voxel that is specific to a fiber population. That even along the same pixel, which is these fibers go in the same direction at the local level, is very hard to distinguish which bundle this belongs to. And if you look at this phenomenon all across the Y matter, you'll see that a very large number of pixels contains multiple bundles, not just 2 or 3, but a very high number of them assigned to it. So this makes it so that these tractogram filtering methods cannot be used on bundles at the current resolution of clinical diffusion MRI. But this doesn't mean that there's no filtering methods designed for bundles. I myself have looked into this along with many others. And one key assumption that we have to make when doing bundle filtering is to assume a kind of coherent structure that the input bundle has, and that it also contains a certain number of spurious streamlines. And the goal is to remove them to kind of preserve that coherent structure of the bundle. And you can apply different operations on these, such as smoothing to help with removing these outlier streamlines. This is a method that's available in DSI Studio, which remove streamlines that's going through these very low density voxels. And these two methods are very interesting where they project these streamlines into this low dimensional space using dimensionality reduction methods such as t-SNE, UMAP, or even a variational autoencoder. And they can then perform this filtering in the low dimensional embedding space to get these very nice output bundles. So while these filtering methods are very nice, they do focus on sort of mostly the correctness scale, like we mentioned, try to remove these invalid streamlines from the bundles and preserving the valid streamlines. So this correctness can be evaluated at the streamline level, where each streamline gets labeled invalid or valid. But another scale that we should be looking at when it comes to bundle evaluation is also coverage, where you can have very underestimated bundles where they're very thin or sparse, where they can be very thick and full or even overestimated. In applying these filtering methods indiscriminately to all the bundles that could lie on this quadrant. For instance, like this bundle shown on the bottom right here, may remove some of these valid connections were the bundle not so thin in the first place. So there's not a lot of methods that really look at the coverage scale, which is one of the reasons that we developed SDNorm. And another reason for looking at bundle coverage is also because it has implications for tractometry. So if we think about bundle coverage, some metrics that can be used to, evaluate that include things like dice overlap, whether a bundle like overlaps with like an Alice bundle. And then if we take it one step further, we can also think about it in terms of the distribution of streamline density. So where the streamline lies in respect, to the whole brain. And that along with bundle coverage, is going to introduce, certain biases. It's going to affect where the core or the centroid of the bundle is. And these are used a lot of times in many tractometry pipelines. And if you think about the tractometry pipeline a bit more, where we're projecting these 3D scalar maps onto the bundles, we're not just projecting them onto streamlines, but we're also projecting them onto every vertex on every streamline. So it's also very important to think about streamline point density, where having different density points is going to introduce certain bias and repeated sampling that could affect the stability and the variance of the resulting microstructure profiles. So streamline density is affected by a number of reasons. In areas where there's more fiber crossing, where the underlying structure have very high curvature, these areas can be harder to tract and may have a lower streamline density. Um, and it's going to also be affected by the seeding strategy in your processing pipelines and the quality of the ODFs, which can be, affected by all the reasons that we talked about earlier that come from SAY and scanner and protocol. So instead of trying to, I think, harmonize tractography directly by controlling for all these effects, we can sort of think of streamline density as sort of a proxy here, where we know that all these side effects going to affect the streamline density, and this and streamline density will also affect our downstream tractometry analysis. So what if we try to kind of control for streamline direct? What if we try to control for streamline density directly somehow? And this is one of the other motivations for our work on streamline density normalization, or SDNorm, that we'll talk about now. So SDNorm is actually heavily inspired by the SIFT method from MRTrix, but instead of trying to match the streamline density from a whole brain tractogram to match the underlying FOD signal, we want to match the streamline density of a given bundle to a template density map, and hence why the name is called normalization. So we do this by first computing this template map from a reference group. In our case, we use 200 subjects from the Human Connectome project, the aging cohort. And then we used a linear model to calculate the streamline weights that we'll talk about in a little bit. So this step is to ensure the distribution of streamline density matches to this template density map. And then this is followed by a streamline pruning procedure where we set a cap or constraint on the streamline point density. So looking at the streamline weight calculation, given a subject bundle and also a template map, we first, assuming these must be in the same space, we first calculate the streamline voxel adjacency matrix, which indicates whether the streamline passes through these given voxels. And then we also represent the density map values as a vector. And then we minimize this objective function, which is the least square with L2 regularization with some of these scaling factors. These are also inspired by the SIFT method, which you can find more details in our paper. And the result here is we get the streamline weights, and these weights will basically represent how well each individual streamline matches to this template density map. So after we get this weight, these will be used in our next step, which is pruning. And we use the constraint on streamline point density. So streamline point density can be represented as the total number of streamline points per unit voxel. But if we approximate the total number of streamline points as the number of streamlines times the number of points per streamline on average, which can also be approximated by the bundle length divided by the step size, we can get this new formulation, is what we call the effective streamline point density. And this property makes it easy for us to do this pruning procedure. We're given a bundle or a collection of streamlines, and then the streamline weights we got in the prior step, and a target ESPD value. We can then perform pruning, where at every step we calculate the number of streamlines we need to get to this target ESPD, and we keep the top end streamlines with the highest weights. And we repeat this procedure until we get to the target ESPD value. And here we're showing some of these different values and how ESPD value can affect the resulting bundle. So I do want to highlight that this ESPD, the target ESPD threshold, is sort of an upper cap set on how many points you can have per unit voxel on average for a given bundle. But this is not a property that we enforce everywhere on the bundle. So you can see from this point representation of this bundle that in some areas it will be more sparse, where the core will appear more dense. It's because we're not enforcing every voxel to have, like 2 points or 8 points per unit voxel, but this is a property you calculate on average. So this is a point of using the streamline weights, because we want this pruning procedure to be informed by, which streamline actually follows the template density map a bit more, and use that way to encourage where to prune and where to keep. So we test this procedure on 140 subjects from the ANI phase 3 data set. across 7 different protocols. And within each, we match by sex and diagnoses. And then we perform a localized bundle tractography or LBT method, which we'll talk a bit more later, to extract these 10 different tracts. And the idea here is that we track only for a given bundle using an atlas bundle as a constraint, as seeding, stopping, so that it's easier for us to alter the parameters for different bundles and different settings without having to generate the whole ring tractogram every time. And in particular, we perform 2 different experiments where we use the same input, process, diffusion image, the same pipeline, with only 2 alterations to the tracking parameter, which is the number of seeds and the step size. And these parameters will directly affect how many streamlines and how many streamline points we get in the resulting reconstruction. So in run A, we set A seeds per voxel with 0.5 millimeter step size. And with run B, we set 12 seeds per voxel with a 0.2 millimeter step size. So from this, you can see you can kind of expect run B to have more streamlines and more densely packed streamline points. So on both these runs, we then do tractometry where we create these along-tract mean FA profile using BLAN. And then we run SDNorm on both these runs with a target ESPD value of 8 and also resample step size of 0.5. So our first evaluation is, very directly looking at how SDNorm changed the streamline density. So here, because run A and run B were extracted from the same data, we expect that they to follow a similar distribution. So if we plot run A, which is shown on the x-axis here, against the run B density map values in this QQ plot, before and after SDNorm, which is shown in different colors, stratify the protocol, we can see that the red scatter plots follow this very steep slope, which indicates that run B has consistently higher streamline density across the entire distribution compared to run A, where after running SDNorm, we see that the blue scatter plots follow this identity line a lot more, which indicates that run A and run B have similar distribution of the density map values. And comparing this across different protocols, you can actually see that the P33, which is a Philips scanner protocol with 33 gradient directions, have kind of markedly low peak density map values. But because these are very low, which is very likely under the target ESPD value of 8, we actually don't further prune this, which is why this line actually don't follow like this identity line as much. But for all the other protocol, we do see this kind of change in slope as well as kind of the peak density. And one other experiment that we did, unfortunately, this one didn't make into the official paper. This actually came from a reviewer comment asking whether SDNorm was removing meaningful subject variability. What we did was we plotted the same plot, aggregated overall protocols, but we performed the Z transform within each subject bundle density map. And then we can see that the density map for run A and run B before and after running SDNorm are actually very similar, which indicates that there's sort of this global scaling bias that's introduced by altering, C's and step size that SDNorm is correcting for. But if we look at the spread of these scatter plots, we can see that the subject variability is actually preserved quite well with SDNorm. And our next evaluation is looking at the along-tract FA profile, where we calculated the difference in FA profile between run A and B. And we saw that FA difference was significantly reduced with SDNorm across all the 10 bundles that we have tested, which indicates that it can improve the reproducibility of the along-tract tractometry profiles. And we also did an evaluation of quality control, and this really came from when we were evaluating the model fit during the streamline weight calculation. We noticed that the model fit R2 was actually a very nice metric for QC. So if we plotted this along with also the correlation of density map values between the subject and the template map, both of these can separate the bundles that pass QC versus the one that was marked as poor and fail. So if we apply some of these R2 value, the bundles with very high R2 and very low R2, we can see that the one with very low R2 typically are very thin, very sparse, they're underestimated, whereas these bundles tend to be very more, a lot more larger and fuller. So we are actually currently using these two metrics in our current QC pipeline for bundles, which we will talk about in a little bit. So some notes on using SDNorm. First, I want to stress that this method was designed for tractometry analysis and it was not designed for connectivity analysis. So please do not use that if you want to do connectivity, and do use methods like sift or commit if you are doing connectivity analysis. A second is that SDNorm is not harmonizing tractography by design, but as you mentioned, it's sort of operating on this proxy of streamline density. So it may appear that SDNorm has the effect of harmonization, but it is not trying to remove these side effects in the first place. And lastly, the SDNorm is only downsampling, but not upsampling the bundles. So we give users the choice to select different ESPD values. So you can select this value to be higher to allow for less aggressive pruning, and higher if you, or lower if you want, more aggressive pruning. So we do encourage users to manually screen bundles with very low ESPD and R2 values, and also to create your own study-specific template maps using whether that's a subset of your study cohort or a demographically matched reference group. So SDNorm is available as a Python package, which you can install here. And we also provide some test data for you to try out as well. But next, I want to go over some of the pipeline that we currently use from bundle segmentation and QC. And hopefully that can provide some guidance how you might want to run tractometry analysis. So earlier I mentioned we used a method called localized bundle tractography to generate the bundles. And we use this method because we were able to generate bundles without whole brain tracking. And this was very attractive for doing tractometry because there are times that you might not want to generate all the bundles. You might just want to, get one or two tracts to analyze them. And maybe sometimes you want to tune parameters for different tracts. So there are methods that already do this very well. For instance, the automated fiber tracking methods from DSI Studio, and also this bundle-specific tractography method from the Sherbrooke Connectivity Lab. So our method is actually very inspired by these two methods with some slight modifications, which is shown in this very complex diagram. But the main idea being that we want to use the Alice bundle to constrain our seeding, our stopping, and filtering. So the way we do that is by warping the Alice bundle, whether in the streamline version as well as the binarized density map or mask version to the subject space, and using that a dilated version of that to constrain where seeding and stopping happens. So shown on the right two tracks here, we want seeding to start. We want tracking to start in one of these endpoint region and end in the other, while following this trajectory that's constrained by the Alice bundle mask. And I want to highlight specifically one decision that we made for this pipeline that was quite specific to tractometry. Is we had this subject that was very cropped around the cerebellum, and we see this a lot in a lot of clinical data sets. So during the last step of filtering, where we use the Alice bundle streamlines warp to the subject space, here we're overlaying the subject's tracking mask on it. If we keep the Alice as is, uncropped, and we use these streamlines to kind of select the subject streamlines, we don't end up with a lot of streamlines. But if we crop the Alice accordingly to the subject, the way the subject brain is cropped, we end up with a lot more streamlines. So while this was a better result, in some cases, this is kind of considered, anatomically incorrect because this is not really a cortical spinal tract anymore because it doesn't end in the brainstem. But this was a decision we were able to make because we knew that we were doing tractometry analysis where we cared more about these along-tract alterations, even though the bundle is a little bit cropped. But also because our downstream analysis method, such as Buang, was able to accommodate where your tracts may be a little bit shorter than others. So it does not affect how the segments were created to establish that correspondence across subjects. So this is one example I want to highlight to say that a lot of these methods should be designed with the downstream analysis in mind, but in a decision that is right for us doing tractometry is, in this case, is definitely not correct if you're doing connectivity analysis. So I do want to kind of make that comment here. And when it comes to QC, we also mentioned this a little bit prior. A couple of steps that we use for QC includes things like heuristic space screening, where you can set these rules like all subjects should have at least half the bundles and each of them should have at least 20 streamlines. So these heuristics can be designed based on different studies that we do. And then we also do a group level QC where we compute these different group average density map that we've discussed earlier. And calculating metrics like density correlation or the R2 score from SDNorm. And then we can use outlier removal methods based on these measures to do a group level QC to flag subjects who are outliers. And for the subject that were flagged in the prior two steps, we can also do a subject level QC, whether through the registration or by generating these 3D bundle visualization figures in this mosaic form, where we can very quickly, screen a subject. So speaking of bundle visualization, I've been a long-time user of the DiPi package as well as Fury, which is the visualization software created by the DiPi team. So over the years, just kind of due to my own need of having to create a bunch of brains, for papers and whatnot, I have created a command line interface that generate these figures, and this is currently available as a Python package called TRK Snapshot. So if you are interested in generating these figures, whether that's, creating custom colors, projecting these FA-DTI maps onto them, or creating these along-tract segments with statistics projected onto those, or even you can overlay a mask as well, do check out our package here. It is one command line interface, and feel free to reach out to me with any questions about this. And lastly, I do want to give a little sneak peek is we've discussed SDNorm, we've discussed, TRK snapshots, as well as many other methods like the localized bundle tractography. We are planning to release that as a toolbox, whether I think soon next month. And this toolbox will have capabilities such as doing QC screening and QC metrics, along with the localized bundle tractography, with SDNorm, with visualization, and it will also support doing bond tractometry. So if you are interested in this, do follow me on socials to hear about it when it comes out. And lastly, I want to thank you to my advisors, especially Dr. Paul Thompson, my committee chair, Dr. Nella Jahanshah, and as well as Dr. Branch Chandio, who is now starting her faculty position in West Virginia University, which is very exciting. And also all of my collaborators and co-authors. Yeah, so we do have this GitHub repo here, which you can clone. And I was mentioning some of the, we provide some test data here for you to use. So I don't think we have time to fully run these commands, but if you clone this repo and copy these commands, you should be able to get these outputs. So just to explain a little bit on what these outputs are. So what you need is a given input bundle, some template map, which we have provided here. And these two need to be in the same space. So here we provide them in the MNI space, but it's also preferable to do it sometimes in the subject space by warping these MNI templates to the subject space, because the templates, the MNI space typically have images, for instance, in 1 millimeter. So it creates a much bigger streamlined voxel matrix. So if you warp that to the subject space, the matrix will be a little bit smaller and the computation may be faster. So if you look at some of the SDNorm, it will output the prune bundles, which is the output bundle that you get from SDNorm. And then it will also provide indices, which is just indices of which streamline in the original bundle was selected in SDNorm. And we also provide the weights in this weight TXT, which you can specify. And this is the weights we'll use in pruning. And we also provide a report with metrics such as, the the model fits metrics like R2, MAE, and RMSE. We also provide the different scaling factors that was used during optimization, as well as like the initial ESPD value, the final pruned ESPD value, and how many iterations it was used to prune. Okay, so I may have time to maybe show some examples of the visualization software that, if anyone was interested in that. So we have this command called trk snapshot, which you can install with pip install. And it's a command line interface with a lot of different parameters, which I won't go over all of them. But it's in some ways, you can do that. It's by supplying your input bundle, in which case we have the arcuate. And we can also provide a glass brain. And if you want to add a mask, you can also specify a mask. For instance, we have our template mask here. And if you want to supply like multiple template masks, you can also do that. For instance, let's try the CST one. And you can specify show to show the interactive window, or specify the output image to save that as a PNG file with some automatic cropping. So here we're showing one example, and this is all made with Fury, which is part of the DiPy package. And you can see we were able to visualize the arcuate with the template mask shown as like a binary mask, as well as a CST mask in the back. We can also do different colors with that. Excuse me. For instance, if I wanted to arcuate vesiculus and CST, you can also specify color, like if you want to show it as red and green, or I guess typically you show them in like green or blue, but let's just specify 2 different colors. Excuse me, we'll take a little bit. Oh, I don't think I activated my. So we can also show this with the glass brain, for instance. So we can also see from the earlier image, our test image was able to be saved, in this PNG format. So if you want to drag the camera direction in different directions and then click X, it will save it in the camera direction that you have dragged into. And we also provide automatic cropping so that all the white space will be removed, so you can paste this directly into your figures. And here is this interactive window, the command we were running earlier, where you're able to visualize these bundles with different color options. And you can also save that into an output file as well. So we do provide that in this GitHub repo, which you can pip install directly to generate these figures.